This is the only company that I have collaborated with, truly collaborated with in this type of a fashion where there's an ongoing back and forth about science discovery that actually goes over years. I work at the Salk Institute where I am a professor and I run a research lab there, and I am also the director of the Salk's NCI designated Basic Research Cancer Center. My laboratory primarily does basic cancer research, but we also do research that relates to metabolic diseases and fundamental cell biology. One of the specific methodologies that cell signaling has pioneered that has really helped advance some of our studies grew out of studies that we had started in my research lab when it was just starting as a fledgling lab. We had identified an optimal substrate sequence for our kinase AMPK. Partnering with cell signaling, we were able to generate an AMPK motif, an AMPK substrate motif antibody, which then, from years of further advancement by cell signaling, was able to become a highly refined, highly purified, and enriched affinity tool. Using that tool then, we were able to profile tissues, cells under various conditions, plus or minus activators of the pathway, with or without a genetic perturbation. And that idea is the idea which cell signaling has adopted broadly as a platform called PTM scan. That ability to profile across conditions with phospho-specific tools, with acetylation-specific tools, with ubiquitin conjugated-specific tools, allows researchers the ability to really hone in on the specific post-translational changes and the protein level changes that occur in two highly related samples. What is the difference between lung cancers with this particular oncogene mutated versus a different gene mutated? It's a platform which enables effectively one to look at the systems biology of a cell, of a cancer, and to hone in on specific targets that may make the difference and may in fact be the target for the next therapeutic around the corner.